In this video, we'll discuss the insulin transduction pathway. Let's suppose you just had a meal rich in carbohydrates. Glucose is absorbed through the intestine into your blood. The blood glucose levels will begin to increase. High glucose stimulates the beta cells from the pancreas to release insulin. Insulin binds to the insulin receptor. Insulin receptors are present on the surface of target cells such as hepatocytes, muscle cells, adipocytes, and even on neurons in the mammalian brain. This blue rectangle represents a target cell for insulin. This is the capillary that feeds this cell. The insulin receptor is a dimer made of two extracellular alpha subunits and two transmembrane beta subunits. Disulfide bonds connect the alpha and beta subunits, as well as the two alpha subunits, to maintain the receptor as a dimer. Insulin binds into this cavity created by these two alpha subunits of the insulin receptor, which face the extracellular environment. Now the alpha subunits move closer together and lock the insulin between them. This also causes the beta subunits to move closer. This results in a conformational change of the protein, which activates the tyrosine kinase domains on each beta subunit. A tyrosine kinase is an enzyme that phosphorylates tyrosine amino acids on target proteins and enzymes. The tyrosine kinase activity causes the autophosphorylation of several tyrosine residues on the opposite tyrosine kinase domain. The process of phosphorylating each other is called cross-phosphorylation. Both of these beta subunits are phosphorylated in the presence of ATP. The phosphorylation of three residues of tyrosine is necessary for the amplification of the kinase activity. The tyrosine kinase domains can also phosphorylate other proteins. One of the phosphorylated tyrosine residues on the insulin receptor, specifically this one over here, attracts a protein called the insulin receptor substrate 1, or simply IRS1. IRS1 gets phosphorylated by the tyrosine kinase domains. The phosphorylated sites on IRS1 serve as attachment points for a lipid kinase, called phosphoinositide 3 kinase, or PI3K for short. Lipid kinases are kinases that phosphorylate some type of lipid molecule. In our case, this lipid molecule is called phosphatidylinositol 4,5-bisphosphate, or PIP2. PIP2 is located in the cell membrane. You can see it right here. PI3K phosphorylates PIP2. The addition of phosphate converts PIP2 to PIP3. Here I'll show the full chemical reaction of the conversion of PIP2 to PIP3. PIP3 stands for phosphatidylinositol 3,4,5-triphosphate. PIP2 gets phosphorylated by the PI3K. Number 2 in PIP2 means there are two phosphates in the molecule. Number 3 in PIP3 means there are three phosphates in the molecule. The additional phosphate will be added right here, at the third carbon. Now back to the cell membrane. Newly formed PIP3 can move along the membrane and serve as a recognition site for an enzyme called 3-phosphoinositide-dependent kinase 1, or PDPK1 for short. PDPK1 phosphorylates the inactive form of protein kinase B, also known as AKT, and now you can see the chemical reaction. The phosphorylation activates protein kinase B. Here you can see a vesicle with glucose transporters embedded in its membrane. These glucose transporters are called GLUT4, which stands for glucose transporter type 4. Activated protein kinase B, or AKT, phosphorylates a protein called AS160, which stands for AKT substrate of 160 kilodaltons. Here you can see the chemical reaction. This phosphorylation inhibits the GTPase activating domain associated with AS160. Now, let's take a look at the AS160 before the reaction, when the GTPase activating domain was still active. In the activated state, the GTPase activating domain is able to activate an enzyme called GTPase. The GTPase binds to GTP and hydrolyzes it to GDP. Now let's look at the phosphorylated AS160 after the reaction. The GTPase activating domain is now inhibited, so there's no one around to activate the GTPase, which is located in the same place. If there's no one to activate it, that means the GTPase is also inhibited. As a consequence, GTP is not hydrolyzed to GDP. I didn't mention that this GTPase is part of a protein called Rab. Rab is the next protein in this cascade. You can see it in pink. I've already explained why GTP is not hydrolyzed to GDP. So, to summarize the big picture of this process, Rab changes from its GDP-bound state to a GTP-bound state, which activates it. Activated Rab gets attached to this vesicle and will carry it to the plasma membrane. The vesicle will fuse with the cell membrane. GLUT4 transporters will translocate to the cell membrane. Right now, we're in the fed state. That means our blood glucose levels are high, as I previously told you. I will denote glucose with this capital G. These transporters will perform the uptake of glucose from the blood into the cytoplasm. Now the glucose is inside the cell. Let's make some room. Protein kinase B has other functions besides those mentioned above. You can see how protein kinase B phosphorylates specific enzymes. These enzymes can phosphorylate or dephosphorylate specific metabolic enzymes, which can stimulate or inhibit different metabolic pathways inside our cells. For example, these enzymes will stimulate glycogenesis. Glycogenesis is the formation of glycogen from glucose. The idea of this process is to store as much glucose as possible inside our cells. These enzymes will also inhibit glycogenolysis. Glycogenolysis is the opposite of glycogenesis. It's the breakdown of glycogen into glucose. We don't need this, because we want to store glucose. These enzymes will also stimulate glycolysis. The idea is to eventually make ATP. They will also decrease gluconeogenesis. Gluconeogenesis means forming new glucose from non-carbohydrate carbon substrates. We already have a lot of glucose, we don't need more of it. These enzymes will stimulate the synthesis of fatty acids from non-lipid precursors, and they will stimulate lipogenesis. Lipogenesis means the conversion of fatty acids and glycerol into triglycerides, also known as fats. The idea of this process is to store energy in the form of triglycerides. The enzymes will also decrease lipolysis. Lipolysis means the breakdown of fats into glycerol and 3 fatty acids. Now let's move on to the next function. You already know that insulin can stimulate the translocation of the GLUT4 transporters from an intracellular compartment to the cell surface. It turns out that in the muscles, insulin can also increase the expression of numerous amino acid transporters. And you can see them in the cell membrane. Amino acids will start flowing into the muscle cells. That's how insulin can increase amino acid uptake in the muscle cells. Now that we have a lot of amino acids inside the cell, these metabolic enzymes will stimulate the synthesis of proteins. These enzymes will also decrease proteolysis. Proteolysis means the breakdown of proteins. Insulin can also decrease autophagy. Autophagy means the degradation of damaged organelles. Postprandial levels of insulin inhibit autophagy completely. These are the main functions of insulin inside our cells.